This is the second of a series of videos detailing how to perform ChIP-seq analysis using Galaxy. In the first video I described what ChIP-seq analysis actually is, and also gave a brief introduction to Galaxy itself. In this video I'm going to download some data that I've pre-prepared so that we can carry on with the ChIP-seq protocol. So to download the data we need, we can go to the Shared Area tab at the top of the page and go to Data Libraries. Now on this example there's probably lots of directories that you won't be able to see just because I'm a member of the core facility. What you should be able to see is one called Workshop Data. So we can see here there's one called ChIP-seq May 2013. This coincides with a training session I gave in May. But what you can see is there are two files, ChIP chromosome 1 SAM, input chromosome 1 SAM. A SAM file is a format of file that is generated by modern mapping programs. The ChIP is the sequence reads from the sample from the chromatin immunoprecipitation, so it's the enriched DNA fragments. Now the input is something that you need to go along with the ChIP sample, and typically this is the DNA that goes into the ChIP protocol but is not enriched using an antibody, so it's basically just random fragments of DNA. We can download both of those by clicking on this button at the top that gets all files from this section, or you can click one at a time. When you've selected the ones you want, you can import them into your current, well, I recommend a current empty history. So press go. Okay, so to go back to the analysis area we can click on Analyze Data at the top. And here we can see two files have indeed appeared in our history section. So while I think about it, I'm just going to discuss why it's best to have an input sample alongside your ChIP sample. This is because during the library preparation step you have to amplify the DNA that goes onto the sequencer. And during this step certain areas of the genome preferentially amplify to other regions of the genome. So if this is left unchecked you'd be left with lots of false positive peaks in the peak calling step. So by using an input sample, which is basically just random DNA that goes into the ChIP protocol, you can subtract away these false positive peaks from those gained by the ChIP sample. Okay, so before I carry on I'm just going to give this history a name. You can do this by clicking up here at the top right hand side where it currently says unnamed history, and I'm going to call this ChIP-seq MM9 chromosome 1. That kind of gives a brief description of what this analysis is about and might allow us to find it in a future date. Okay, so so one thing you have to make sure you do is actually press return. ChIP-seq MM9 chromosome 1. Hit return, don't just click away from this box, otherwise it won't save it. So there we go, that's the name changed. Okay, so we've got our two input files. Sorry, I shouldn't say that. The two files that we're going to use for the ChIP-seq analysis. One being for the chromatin IP sample for our transcription factor, and the other which is our random DNA input. Okay, so let's crack on and do the peak calling step, which uses the tool MACS or MACS. So I'm just going to find this, I'm just going to use the search tool MACS, and here we can see under NGS peak calling there is a tool called MACS 142, and this is the version 1.4.2 of MACS. So we can click on that, and it brings up in the middle portion of the screen all the options that you can select to run the program. Just a note on MACS itself, it's been around for a good couple of years now, and it's probably one of the most widely accepted peak callers in the field. This version is the current stable release, 1.4.2. There is another version, version 2, but this is still in what is called an alpha release, which is a kind of development version. However, more and more people seem to be going to this new version, as it is probably, well, let's say it's the best it's going to be at the moment. 1.4.2 is fine for most jobs though. Okay, so you can give the experiment a name. So I'm just going to alter this. MACS 142 NM9 chromosome 1. For this section here, paired end sequencing, we want to keep this single end option. This is because our reads are from fragment sequencing, single end fragment sequencing, and not paired end, which is the other option. So we can leave that the same. Our ChIP-seq tag file or sequence read file is history item number 1 from our list, and the ChIP-seq control file, that is our random DNA input file. Okay, so for this option, effective genome size, this is required by the program when generating its statistics. Now by default it gives a value in base pairs for the human genome. This is specifically for the human genome. The effective genome size is, as a rule of thumb, 70% of the available sequence, because quite a lot of the sequence can't actually be sequenced by today's sequencers because of repetitive regions, for example. So at the moment this is 2.7 billion, that's specifically for human. We want to change that for mouse, which is, if we look at this little line here, 1.87 billion. So it's 187 with 7 zeros. 1, 2, 3, 4, 5, 6, 7. We can just double check by counting back along here. So it's 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1 billion. So that's fine. Okay, the tag size is the length of the reads from the sequencer, which is 36 base pairs. The bandwidth is the mean fragment length prior to library preparation, i.e. you giving your DNA samples to the sequencing facility. Generally this is around 200 to 300 base pairs, but for this particular sample I'm using 200 base pairs. The p-value cutoff, this is a reasonable default. It's quite stringent, so you can see what results you get using this default. If you're too few or too many, you can change it up or down accordingly. So 1e to the minus 5, if you want to make that less stringent, you can change it to 1e to the minus 4, which is equivalent to 0.0001. Okay, this option here, you can pretty much leave that. This is to do with predicting what the fragment reads are in the sample after sequencing. And we want these two samples selected because we want the detailed output for downstream parts of the analysis. We also want to keep the wiggle output file. Now, a wiggle file is it's a read coverage file, so on a genome browser you can see which parts of the genome have more reads or fragments than others. This is kind of a typical profile you see for this sort of analysis. We want this again for downstream analysis, so we want to save that. Okay, we can keep this resolution, we can keep this as default, this is fine, and the rest of this, unless you know what you're doing, you should keep these as the defaults. Okay, so having selected our parameters, we can then click execute, and this will put our job into the Galaxy queue. Okay, so you see at the moment these are clocks which are they're waiting for the jobs to go into the queue, and in a minute these will turn into, here we go, into blue rotating circles. This means that the jobs are actually running. Each of these items in the history bar, these are different outputs from the MACS analysis. Incidentally, the names of these things look quite cramped, so you could actually change the width of this history bar using this this little tab at the bottom. You just click on it, hold, and you can drag it to make it bigger. The same goes for the left hand side as well. So while Galaxy is running our MACS peak calling step, I'm just going to talk about MACS itself a moment. At a very simple level, what it's going to do is scan through the genome and look for areas where there are many more reads from the ChIP sample compared to the input sample. Now, MACS doesn't just use the 36 base pair reads in this analysis. What it does is it tries to simulate the actual fragments that have been sequenced in the sequencer. Now, this is just a little overview again of the ChIP protocol. So you've got cross-linking of proteins to DNA, some of which contain the protein of interest. These are sonicated into bits, so these are our DNA fragments, some of which contain the protein, some of which don't. Using a specific antibody, the pieces of DNA that contain the protein, in this case the green circle, are enriched, and the pieces of DNA that don't contain the protein are washed away. Now, during the actual sequencing process, after amplification, you have these fragments, and they're usually sonicated to around 200 to 300 base pairs. These are often sonicated again or size selected by the sequencing facility. Now the sequencer itself is only going to be sequencing 36 bases from each end of the fragments that go into the sequencer. Obviously this is a lot less than the entire length of the sequence. So for example here you've got a fragment and you sequence in from the 5' end of the forward strand and the 5' end of the reverse strand. So if we map these reads onto a genome browser, which is what this screen is here, all the blue, the little blue areas here are the forward strand mapped reads, so the start of the fragments, and the red ones correspond to the reverse of the reverse reads to each of these fragments. So if we look at these forward and reverse reads on their own, you can see that for one binding region there's this kind of bimodal distribution of reads, these ones corresponding to the forward strands of the reads, these reversing to the reverse strands. So what MACS is able to do is say, well, I can see this distribution of reads, let me guess what the mean fragment size is. So in this schematic on the right hand side, Watson tags corresponding to forward strand, crick corresponding to reverse strand, it looks at these bimodal distributions, finds the center point of each, and then looks at the distance between the peaks. In this case it finds what it calls D, the distance between the bimodal peaks, of 126 base pairs. So with this D value in mind, it takes all these 36 base pair reads and extends them 3' to make them into 126 base pair fragments, and that is what it uses in its analysis. Now additionally, if you have a read, say for example this one, and say this goes from position 100,000 to 100,036, there can only be one read in that position on each strand, so 2 reads in a particular position. There are exceptions to this, but I'm just going to keep this simple at the moment. Now what MACS does then is it takes a non-redundant set of reads, so only what I just said, up to 2 reads in each position on each strand. It throws all the other duplicates away and just uses the non-redundant set in the analysis. Sorry, you may want to go through and listen to that a couple of times because it's not terribly simple. But what it's doing is, by getting rid of all the redundant reads, it's ensuring that you do not get false positive peaks due to areas of the genome that are over amplified. Now of course you do have your input sample to guard against that, but even so you can still have areas of the genome that are over amplified during the library preparation step. Okay, so if we look back at our Galaxy session, we can see that MACS has completed as all the items have gone green, and it has produced 7 different output files. So I'll briefly go through what these files are. We don't need them all in this particular workflow, but it's important to know what's in them. So the one that we're currently viewing on the middle here, the middle section, allows you to see what's in a particular file up to 1 megabyte of data, and it
is providing us with information which is the genome coordinates of the binding regions found by MACS. So we've got chromosome start, end, the name of the region given by MACS, and its transform p-value, the minus log 10 of the p-value. And there are 674 regions that have been found. The next file, item 4, is the coordinates of the summit of the binding regions shown here in item number 3. So this is the coordinate with the highest pile up of fragments. So that's important later on because it pinpoints where the sequence of interest is for transcription factor binding. Okay, I'll just close these to make space. This is the detailed MACS output. I'll just put that in the center. Now this gives us information such as the parameters that we used to run MACS, which is important for knowing what we did later on or in the future. It also tells us the number of reads or tags that were input and the number that remained after the redundant reads were removed. So in this case it's saying that it kept one read at each position, or at least one read on each strand of a position. With smaller genomes and high read coverage, this value can sometimes increase. And also it shows the value of D it predicted, which again is the distance between the bimodal peaks of forward and reverse reads. It's slightly more complicated than how I described it, but that will do for the moment. Okay, so that's the detailed output. I should probably just describe what these different columns are. So here we have the chromosome start and end coordinates of the binding region, the length of that region, the position of the summit relative to the start coordinate, and the number of non-redundant reads that are present in the binding region. This is the transform p-value, giving an indication of how good that peak is relative to its background, and the fold enrichment relative to the background, which is the input reads. Now it doesn't just look at the input reads directly, it uses a special lambda model which is terribly complicated and I can't describe. Basically what it does is it looks at the number of input reads in the nearby area to the binding region and produces a model. All you should really take home from this is that any binding region with a fold enrichment less than 10 is probably going to be a false positive result. It's a rule of thumb, but it seems to work in my experience. The false discovery rate is produced by swapping the chip sample with the input sample. So at a given p-value it looks to see how many more binding regions would be found. As a rule of thumb, I only use binding regions with a false discovery rate of less than 10%. That's quite complex, and I've probably hashed describing that, so if anybody has any questions, then feel free to contact me about that. Okay, we have the peaks that come from the sample swap, so whereby the input is swapped with the chip. These two files are the wiggle files of the fragment coverage, non-redundant fragment coverage over the genome. We can use that in downstream analysis. They can also be uploaded onto a genome browser. However, you need to do some extra work on these files to do that, and you need to be able to host them from a web server. However, if you run jobs through the Bioinformatics Core facility, these will be hosted for you as part of the data analysis. Lastly, we have the HTML input, which you can view by clicking on the iGem. Again, it gives summary information like we saw in the this file here on item 5, but much more detailed output as to what MACS was actually doing. So for anybody who's interested, the build peak model is involved in predicting the value of D. Incidentally, we can get some more detail on the peak model building step, which finds the mean fragment size, by looking at this PDF file at the top, and this shows where all the reads on the forward strand lie on the genome, and where all the reverse reads lie on the genome in the sets of test peaks that it uses to create this model. In this case, the distance between the two bimodal distributions is 142 base pairs. Okay, so MACS has provided us detail on where interesting binding regions for the transcription factor lie on the genome, and in the next video I'm going to describe other tools that can be used to get some more biological detail about the binding regions that we've found, because at the moment it only just tells us where these regions are, but not much else. Okay, so as well, I have produced a Galaxy page that provides extra information on all the steps of the ChIP-seq analysis, and this can be accessed via the Shared Data tab at the top, and then if you click on Published Pages, there is a page called Basic ChIP-seq Workflow, and if you click on that, it will open up a page that has detailed information on each step. Okay, so I'm gonna close this video now, and hopefully I'll see you in the next one. Bye.